Today I am going to talk about what is new and what is coming in PlantGenIE. The Plant Genome Integrative Explorer is a collection of interoperable web resources for searching, visualizing, editing, analyzing, and sharing genomics and transcriptomic data for different plant species. Currently, it includes dedicated web portals for in-depth exploration of poplar, Norway spruce, Arabidopsis, and Eucalyptus. Let's look at the high-level structures of the PlantGenIE system. We get data from different data sources around the world, as well as data generated within UPSC. Then we use different types of parsing scripts to convert those data into suitable formats and store them in here at Umeå University. We also have an API. API means Application Programming Interface, which works like a middleware or the communication medium between the database and the user. We have different web services or API for different categories of tools and databases. Those who are familiar with the programming know what's the meaning of API or how to use a web service. However, I will show you some example how to use the API using command line. As an example, if we give this command, we can get the expression value for given gene ID. Here I gave Eucalyptus gene ID to get biotic stress gene expression values. We can get best diamond, which means faster version of blast for given Arabidopsis IDs. We can get Go, FEPAM, KIG enrichment results for given gene ID. Here I use again Eucalyptus IDs. You can read more about our API on api.plantgenie.org website. However, I am sure many of you would like to access our PlantGenIE databases through graphical user interface, not using command line. Therefore, we develop different types of tools. We have different categories of tools like expression, genomes, comparison tools. We also have tools for searching genes and annotation. Also translation between different versions. As an expression tool, all our X tools like XNet, XImage, XSeedMap, XPlot and XMatch are categorized as expression tools. When tool, enrichment and complex are considered as a comparison tool. Rest of other tools like BLAST, chromosome diagram, JBrowse are considered as genome tools. This is one of our expression tool called XImage. This shows us pictographic view of expression data across different tissues. In this picture, we use from Zostera marina tissues for visualizing expression variations. When I click the gene ID, we can see the changes in different tissues. This is the same way that we use in API. As you see in the above, we have the API request. The similar thing we do using graphical user interface. So I think using a web interface tool is much easier than the command line to access the PlantGenIE database. I think many of you are familiar with the PLASA. PLASA is a plant-oriented resource for comparative evolutionary and functional genomics. Currently, PLASA includes more than 70 species. The PLASA platform consists of multiple independent instances focusing on different plant grades. We have installed a custom version of PLASA for UPSC with our UPSC STT VIB project. As a result of UPSC STT VIB project, we use parser pipeline to generate and store ortholog information within PlantGenIE. So we can integrate parser ortholog information to move in between different gene sites. We are planning to integrate comparative evolutionary and functional genomic data into PlantGenIE. In the future, we can make better visualization tools. Here is an example of how can we integrate parser information into PlantGenIE. We have different gene sites, but we do not know how to move one from one species to another. We can connect different gene lists using cross-species ortholog genes. So we can use parser ortholog information. Parser has four different methods to store ortholog information as shown here. We also calculated BLAST and reciprocal BLAST from UPSC bioinformatics platform. Therefore, we can create different cross-species gene lists to move between different gene sites. Here is a screencast showing how this function works in UPSC. As you see here, we just saved lag case gene into UPSC gene list. Now we are going to create a cross-species gene list for poplar, spruce and Arabidopsis using best diamond hit. When I go to one of the gene sites, I can be able to see this cross-species gene list. This is PopGene. We can also create a cross-species gene list using complex. The complex is a comparative analysis of plant expression network, which uses functional orthologs or express log information to visualize network in different gene sites. This information can be used to move from one species to another. We generated a lot of RNA-seq data using UPSC bioinformatics pipeline, and we need to visualize them in PlantGenIE. We have different expression visualization tools. As I mentioned earlier, XImage provides an intuitive pictographic view of expression data across diverse range of PlantGenIE datasets. It helps to visualize differential expression levels across multiple samples using different expression values. This example, you can see the absolute TPM value generated using our pipeline. When we shaded the sample based on absolute expression value, we can't see any significant color differences between tissues. Therefore we use relative mean differences to amplify the expression differences in different samples. Here you can clearly see the significant color changes in shoot tips and mature leaves, where mature leaves get low expression values and the shoot tips gets high expression values. XImage is a nice tool when we have genes of interest to visualize expression values. But how about the other way? Which means we could color the samples and get the relevant genes. We have a tool called XMatch. This is an ideal tool to find tissue-specific promoters. Here is an example demonstration showing how the XMatch tool works. Imagine if we want to find genes that are highly expressed in flowers. The user can drag the colors from the color legend and drop into the sample of interest. When we click the Find Gene button, this will find 62 highly expressed genes in flowers. It is possible to color more than one sample at a time. As you see here, the user can color the sample using the color legend. Blue color represents the low expression values, whereas the red color represents the high expression value. Therefore, if you drag color from the color legend, we will get genes that are highly expressed in flower stage 2. First we need to get the maximum values or minimum and minimum value. Then we can create the scale. We can distribute the expression value for different colors in color legend. Finally, we get the genes for selected color. Each color represents the expression range.